I am going to talk about Plasmodium knowlesi. And to start with, I'm going to give you a little bit of a background about Plasmodium knowlesi because it's not a conventional plasmodium. Its main difference is that it's a zoonotic disease, a zoonotic infection. And its primary hosts are macaques, long-tailed and pig-tailed macaques, and therefore that brings an extra dimension to our thinking about the epidemiology. And so as well as that kind of zoonotic infection angle, I think the other thing that I would say is that this is juxtaposed to this morning's talks, where my geographical focus is going to zoom very rapidly down to kilometre scales rather than aggregating data from across countries or geographical areas. So Plasmodium knowlesi was first discovered in the 1930s. And as a simian parasite was shown to have varying levels of virulence in different species, in particular in rhesus macaques it's a lethal infection, in the long-tailed macaque it's not. So it's showing some level of propensity to cause different disease dynamics in different hosts already in our first kind of knowledge of the parasite. It was used reasonably widespread, or it had a widespread use reasonably in the treatment of neurosyphilitics, one of our great innovative medical approaches in the 30s in treating syphilis by using the spikes in fever of malaria to kill the syphilis bacteria. The interest there with knowlesi is that, and this sets it apart from other malarias, is it has a 24-hour life cycle. So the fever paroxysms are reached every 24 hours, whereas the classic infections in vivax and falciparum are thought to be around 48 hours. So that tells you something about the biology of the parasite in that it goes through that asexual cycle much more frequently than falciparum, and therefore, in theory, an uncontrolled infection, a non-immune host, it can rapidly reach very high parasite densities, and you can imagine that has some consequences. As far as human infections are concerned, there were a few isolated cases. Some of them made the news. One was in a CIA agent who was in Malaysia and Borneo. The other big news came out in 2004 when Bal Singh and Dave Conway from the London School described a large foci of naturally acquired knowlesi infections in Sarawak. So molecular techniques, which is the best diagnostics, and I'll show you why next slide, are PCR, molecular amplification of knowlesi-specific genes, revealed that 80% of their infections were not Plasmodium malariae, but were in fact Plasmodium knowlesi. And in contrast to knowlesi, Plasmodium malaria is thought to be a rather benign parasite. It has a 72-hour life cycle, so it reproduces pretty slowly. And this is part of the reason why classically knowlesi was called malariae. And I know you're all budding, aspiring malariologists, but even to those of us who think we know a bit, it is extremely difficult to tell the difference between this ring stage of Plasmodium knowlesi and P. falciparum, and the later stages, including the gametocytes, the transmissible stages, and P. malariae. And so essentially what was happening was a misdiagnosis of P. malariae as Plasmodium, of P. knowlesi as Plasmodium malariae. And so some colleagues, Tim William, Bridget Barber, working out of Menzies and the Ministry of Health in Sabah State here, and the maps do improve, I promise you, working here in a retrospective study, found that 75% of their admissions were actually P. knowlesi. And of these, a high proportion had severe manifestations. And the clinical features are detailed here. You'll notice no cerebral malaria, no sequestration of the parasite in knowlesi. But 11% of all admitted cases and 28% of these severe cases died. And so that is a close to 30% case fatality rate in severe malaria is very similar to that we see in P. falciparum. So whilst there are admittedly very, very few cases, it has the propensity to be a very severe infection when you get it. And again, in Kudat Hospital, which we're going to see and move to in a minute, up here, they found that nearly 90% of all malaria was knowlesi, and that of those cases, 10% were children. And the reason that statement is in bold is because knowlesi is largely considered to be adults who go to the forest and are in an environment where there are mosquitoes that have bitten the macaque and then can onwardly transmit that infection to them. But clearly what we're seeing are infections in children who would not normally be expected to undergo that activity. I think the other important thing to point out is the Malaysian malaria control program have done extremely well over the last few years in removing the two main human malarias in their country. And they have falciparum and vivax are down to extremely low levels. On the other hand, this graphic shows increases in cases of knowlesi in nearly all the districts, bar this relatively uninhabited district over here, of knowlesi in terms of certification. So the number of cases is rising across this particular state. So Dominic presented this morning the 1,000 Genomes Project for Anopheles gambiae. Unfortunately, life is not so easy for the knowlesi vectors. There's a whole set of complexes that have been associated with transmission of knowlesi or implicated. Some are confirmed vectors, others are thought, well, if we caught one, we wouldn't be surprised if it was infected with knowlesi. And one of the kind of interesting things and a really important caveat in terms of control of knowlesi is these are located in particular ecological niches. So it's unlike Afrotropical vectors, which will breed pretty much country wild. They have a little bit of specificity in their breeding sites. Here we're looking at the appropriately named littoralis, breeds on the coast, and then we have a variety of others who are found in these either slightly dappled shaded areas in pools by rivers, others that are found in more thickety type forests. It's an incredibly difficult entomological approach. And even some preliminary work that we've done, I'll just wait a minute while you write all those down. This is the kind of entomology that I like to do. You put a cow in a net and then go away, and then you come back the next morning and you can hoover up mosquitoes. But the graphic here, and I hope you can make out kind of even, not necessarily the digits, but the numbers of digits in each one. We've got a couple of different sites here where we are up and down is altitude-wise, so lowlands, highland. And what we're seeing, even within one site, we see great variation in not just the type of vectors we catch, but the abundance of different vectors that we're catching. It's an incredibly complex entomological picture. But it is not just restricted to Malaysia. Malaysia has had most of the cases reported in the literature. And this is Sarawak here, this large red blob, and I think the orange blob there is now bigger than the red blob. This is from when, 2008. I'm fairly sure that those blobs will have overtaken each other. But the other circles represent cases where, or countries in which cases have been reported. And again, I know that there have been cases reported in Laos and I think Cambodia too. The two main confines of distribution of this parasite are really the distribution of the long-tailed macaque, which is the black line, and then the pink line, which I hope you can make out, is the distribution of the leucospirus, what's thought to be the primary vector. The two maps this side are indeed done by Catherine and the group in Oxford, with some of us contributing data and ideas to this, basically showing potential risk for transmission. So we can see it's not purely localised in Malaysia, but this is where the bulk of the cases reported so far have been. As I say, it's very complex dynamics, and this is an oversimplification of a very complex process. What we have essentially are human settlements on the edge or outside of forest areas with some level of plantation, and what we have here are the forests there which the macaques in the grey bars are moving through. And one suggestion is an analogous transmission system to yellow fever, where you have one vector type maintaining macaque to macaque transmission, perhaps higher up in the canopies. Another vector type maintaining macaque to human transmission, as humans will tend to sleep when they go to the forest or be active on ground level. And the big question mark, and one we're concerned about, obviously, is human to human transmission. Not something that I intend to answer unless we go to the pub afterwards. But I think the premise here is what we're looking at is an emerging infection. And a lot of those kind of details about transmission and the exact dynamics of transmission are unknown. And so Monkey Bar is the name of the program that I am running. You can ask me later how we got to that. But what it is a call, an answer to a call from the UK Research Councils, who I think are to be commended. So this is our basic bioscience, medical, environmental and social research councils, putting their money together to appreciate the need for integrated research in addressing some of these emerging infectious diseases. Emerging because of potentially we're living with environmental change, and that means both social change, i.e. increase in population movement, as Andy described, increased urbanisation, or increased human pressure on the environment. And clearly, my previous slide, if you can imagine disturbances to forest environments, that is a credible hypothesis for the increase in spread of zoonotic malaria, knowlesi, in Southeast Asia and in Malaysia in particular. So we have two main project sites. We have Kudat site up here. This is Palawan Island on the Philippines. Highly recommended in Condé Nast if anybody wants to go there. And we are combining all the basic kind of malariametric techniques with some new approaches, and it's the kind of more new approaches I want to share with you today. And so, as I said, one of our credible hypotheses is that changes in deforestation, or increases in deforestation, changes in land use cover have really been driving the likelihood of emergence of Plasmodium knowlesi. And I don't know whether you can see that reference at the bottom. This was a paper published in Science a couple of years ago where they had provided very detailed information and accessible information on changes in forest cover over the last 10 years. And one of the targets of their research was included Malaysia as an area where we know there's been huge environmental change. And that environmental change, to put it not too bluntly, basically involves removing secondary forest and replanting with palm oil plantations. So I'm going to just toggle through a few slides, and what you can see here is the forest cover for the tip of Borneo in the year 2000 is around 86.5%. And we can go through, and you can already see in 2008 it's down to 80%, and there's been quite large tracts removed here. And again, the most recent one that we have is 2012, and we're now down to 75%. So that's a kind of a gross 10-12% move, but actually when you think about that in terms of where that has been taken from, it's almost certainly that this is more localised around human settlements, moving away from settlements, eating into forest. And so one of the first things we did was we collated village level malaria incidence data from microscopy records in the area for which we had the land cover data from. And these are kind of smooth maps of village level projections of microscopy positive and adjusted for, because we started before they had PCR records, but we've adjusted for that PCR confounder there. And what we can see is the clear emergence of some areas of hot spots of transmission, or villages which are more likely to have knowlesi than others, and perhaps none more so apparent on the island at the top here, this is Bangi Island. And as you go through time, you can see this white spot on the south tip here getting larger and larger and larger, and up until 2010, very few cases coming from Bangi. But now, 2011, 2012, we're seeing that, and that has continued into our own surveys, which are ongoing at the moment. So what we're seeing, and slightly paradoxically, perhaps, although not if you think about it, we're seeing, I've missed off a bit there. So we see significant correlations with both proximity to the forest and loss of forest. And so our project is, the actual workings of the project, the day-to-day running of the project, is designed to drill into that observation a little bit more. And so what we have, and what I'll try and describe to you, is a case control study for knowlesi malaria. So essentially, anybody that comes into the hospital is with identified knowlesi malaria, PCR confirmed, is then followed back to their house. And from that same geographical area, we choose 3 other control houses who don't have malaria. What that does, that sample selection of cases and controls, gives us a kind of a spatial area in which we can operate. And about 6 months into the project, we were able to identify areas in Malaysia where we had higher than expected levels of cases, and somewhere where we felt we could focus some of our more detailed work. So we have some entomology ongoing. I'm not really going to talk about that, but I'm happy to take some questions about that. We have some social science. Again, what do people think about this disease? Do they understand the concept of zoonotic? And again, I'm not really going to talk about that except to talk about some of the spatial work. And then the primatology, which is obviously a new concept to me as a conventional malariologist, and how we dealt with that. And I won't talk about phenology, but phenology, for those of you who don't know, is the study of tree types and changes in tree types. And that influences where the macaques might be at any time of the year because they need a food source, and some trees will be fruiting at different times. So this is the basics of the results of the case control study, but as I say, what I really want to focus on in what it's allowed us to do spatially. And so we've had the vast majority of our malaria cases are knowlesi, which is great. We've got these 2 unknowns, watch this space, we're not quite sure what they are. We have the majority of cases are within our expected risk group in terms of age and behaviour, i.e. individuals who will go to that forest and potentially and do their work there. But we still have, again, that figure of 10% relatively small individuals, children, and we have several household clusters of infection to suggest that perhaps knowlesi is not just transmitted in that kind of forest environment, but there may be some peri-domestic transmission. The methodology we published for that in last year. And so what we were able to do was take one area and focus our activities in that area. And to give you an idea of what that is, what we have are primatology transects. This is classic walk up and down a few times every month and see what you can see in terms of sites or evidence of macaques. We have entomology collections performed pretty regularly in selected sites, and the red dots are our cases, and the arrows are our houses also in the village. Okay, so that's the baseline area in which we're working. One of the issues we faced is that actually we had quite a large kind of budget to get our satellite imagery, but any of you who have worked with that before know that it isn't particularly useful in the sense you can only access it at certain times. This is Borneo, this is the rainforest, clues in the name. It's quite a lot of rain and quite a lot of cloud cover. So we wanted to have something that allowed us much more regular and actually in-depth assessments of our work. So we bought a drone, and there it is. This doesn't carry any weaponry. It's quite small, and this is us doing our Top Gun impression. And you know, this is probably the pinnacle of my career, and we've made the front page of Trends in Parasitology. But this is effectively what we've used for our mapping. So it's a Sensefly B. It weighs about 700 grams. It's still classed as for insurance purposes, still classed as an aviation vehicle, and therefore it's very expensive to get insured. It flies at about 400 metres, so you can get it up to 1 kilometre if you want to, and it will give us down to about an 11 centimetre pixel in terms of our ground coverage estimates. So the flight is pretty much pre-programmed. You know where it's going to go, you launch it and you let it go, and you do it zigzags across the area. And so this is just some screenshots of actual flight path here, where it's been, what it's doing over that period of time, and then those data get interpreted. Because if you see it in the air, if you actually look at it real time, there's an awful lot of pitch and yaw, and so actually the vast majority of the cost of this is the software that removes a lot of that and makes the imagery a stable product with which you can work. And so you begin, as it begins to process, you begin to make your underlying map of your surface, and your final product is something like that. So that is the result of about 5 or 6 flights in which we've overlaid and got here a nice 3D kind of image there, which we can begin to work with. You obviously get altitude as well as all your land cover images. So the question is how do we practically use that? So again, this is what we would have done before. I chose my colours there. Well, that is the house of a case. Okay, and so what we're thinking, okay, so we've described these places where these interactions occur. What we've got is somebody's back garden here, probably a bit of cassava, a bit of maize in here. We have a young plantation here, a young bit of palm here, and then this area is secondary forest. What we're now doing is automated land use classification on a detailed level. So we've zoomed out with the same image. We're using e-cognition to allow us to classify land use types into specific strata that we can then begin to work with. So just to take you through, that's the basic image. We then overlay on our secondary forest, which as you saw before was just opposite the house, and there's still some remnants all the way around. And areas where, and these will be kind of borrows or slight riverine areas where they haven't been able to dig out and plant the palm. And then we add on a level of two main crops, this will be coconut palm and this will be rubber. And actually the main crop outside in the larger areas is palm oil. So we can begin to build up this map of our study area. And the other thing that the drone allows us to do, and you have to take my word from it, these are the same pictures 2 months apart. This is not something we can readily characterise and readily get information for with that same timestamp way from satellite imagery. And I'll show you the relevance of this in a minute. So that definitely is, you can just about make along that ridge there, which is going along here. But pretty immediate and catastrophic reduction in, well, change in vector habitats, probably pretty catastrophic for your average macaque who thought lunch was there in late February and came back in April and it wasn't. So one of the key factors here, and a completely new dimension to my work, is actually trying to get an understanding of primatology. And this was a whole different adventure. So we work with the Sabah Wildlife Department because, as you know, Borneo is a major conservation hub, and they are very proud of the work that they do. They have managed to put collars on nearly every animal you can think about: crocodiles, monitor lizards, tiny little nocturnal primates. So this they thought would be fairly easy. We had a lot of preconceived ideas about how we would do this. We would put the collars on females because females are nice and stay with the troop, and males are not, and they wander off. It turns out females don't have a chin, so it kept falling off. So we eventually found that actually males aren't as bad as we thought. We got some collars on males. And the scientific reason for asking this is we want to know where macaques go. We want to know where they spend the night, essentially. But there's also the idea that they are the parasite biomass. They're the natural host for this infection. And it's quite important that we understand the level of infection within that, as that is probably what drives the truth. This actually image is courtesy of Amy, who works in Singapore, and their macaques have very low levels of infection, in fact none. Urban Singapore macaques have no infection. So why is that? And so this is the first kind of feedback from a macaque that we collared on Bangui Island, the one we found a lot of malaria. And two things to kind of point out from here. You'll see from the key that the yellow is the yellow is daytime movement, and the blue buried here are roosting points. And if we're, our conventional malaria might have mosquitoes bite at night or dawn and dusk, that's when we're interested in. The other thing is that you can see that this is also a recently heavily cleared area. Well, perhaps less recent because we've got quite some greenery on the ground here. And the mosquitoes seem to have the macaques seem to have gravitated to their movements around this particular area of clearing. So again, that's telling us that they are responding to, in some way, the fact that their area has changed, their feeding area has changed. And again, so what we have here is a little bit of that site that I described previously, but now overlaid. So just to orientate you, these images are not finalised. We're still kind of patching them together. You can still see some of areas are missing. But this is the road going through. These are the houses on the roads and in the village. And this is our macaques daytime and nighttime roosting before any land use change. And what you can see again, as soon as we see land use change and response to habitat disruption, we've got this big area cleared here, which I showed a snippet of before. All of a sudden the macaques have gone onto the other side of the road. So that's one macaque, we're assuming, representing the troop. And the next month you have, or the next time we flew the drone, you have even more of the area is cleared, and the macaques have now totally moved north. What we know, all the macaques we have so far caught and collared are infected with knowlesi. We obviously can't make any direct implication that they have subsequently onwardly transmitted malaria to humans, but the fact that they are moving, and this is as a result, almost certainly of this, is of a concern for us in our predictions of kind of risk factors for this infection. So one of the newer things we're trying to do, we've just got some data, one slide I was able to change today, is as I said before, we've got one macaque that's collared. We don't really know the size of those troops, and the size of the troop will influence the parasite biomass and the likelihood of transmissibility to any mosquito that feeds on it. So what we did was we got a thermal camera, and these are for London Zoo, not Malaysia, but these are from the Philippines, so this is the camera handheld. So what we're trying to do is remove, that was a hot day, so you can see the boughs of the trees are actually hot there. We create a temperature mask here, and we're able to identify these black blobs are macaques. You have to take my word for it again. Then we stuck that camera on this drone here. The previous drone's fixed wing drone, so flies straight line. This is a copter drone which will fly up and basically do what you tell it. So the next step is obviously to try that in the field, and so whereas we had us looking like Top Gun before, this is a bit like kind of, I don't know, some UFO type movie, because it has to be flown at night. And the idea here is what we're doing is identifying where the macaque is from the collar information. So we have a VHF recorder, we can identify where that is. We then fly that in the forest above there, and we begin to see images here. Again, these are macaques. We know they are not larger primates or warm-blooded organisms because we can identify those. And again, you have to take, this is the most recent flight we had, that white spot there is our collared macaque. So what we're doing now is beginning to develop image analysis process, which are actually based on cells and microscopy, to try and feed this kind of print into our software and identify and enumerate what we think are clearly our macaques, to allow us to build up troop numbers for our models. So, as I said before, I don't really want to talk about the entomology too much, but what I will say is we are using these detailed drone maps and our land use classification to create grids of likely breeding sites. So the idea here is that you're saying, okay, so this is predominant land type, predominant cleared vegetation, or here is recently cleared. That is where we're going to do some larval sampling here, and we will contrast the outputs with larval sampling from, say, which we think is much more secondary forest. Okay, so the idea is to have a directed approach to mosquito sampling to enable us to build these composite levels of information around where the macaques are and where the mosquitoes are. One of the larger problems are the humans, because they're relatively unpredictable. As Andy talked about this morning, an awful lot depends on wealth, and so this part of Malaysia, I mean, Malaysia is a relatively well-off country, and although Sabah is at the poorer end of that scale, there's still quite a lot of vehicles, so there's still quite a lot of movement around. And we really want to get an idea of what people are doing in terms of their interactions with both macaques and mosquitoes. So we use GPS trackers, not collars, on people. And what we have is a select group of people, and we ask them to carry those around, and we ask them to carry those around at certain times of the year because we want to get an effect of seasonality of movement. We also ask them to record when they see monkeys. And this has only really been a problem when they've got a pet monkey. because they just sit there clicking the button outside, which is not helpful at all. So again, as I showed you with the macaque and the collar data, what we can do is we could timestamp this. So what we have here, we have a mother and son. I think, yeah, the mother's in red and the son is in yellow, and the son works at the plantation, and that actual bit there is the area where we had the macaque movement and the clearing from before. So he goes to work in the plantation. So this is over a period of a week. Mum either swims or goes out in a boat. I suspect it's the latter, and does that, and moves down here to the main town, which is down here. But again, what we can do is timestamp that. So that's that same kind of collection of points, but it's timestamped to crepuscular, so between 6 and 6, the likely biting times of our vectors. The big challenge, of course, is integrating this. And I'm not really able to tell you much more about this. As Dominic said this morning, we are also collecting bytes and bytes of data to enable us to do this, but this is some example of what we had so far, where we're looking at the basic land use image, macaque movement and human movement overlaid on the several land use classification types. So our secondary forest, what we can see is actually the most likely points of interaction from here are people going into the secondary forest and encountering macaques there. So that is still the most likely source, at least from this in this particular area. And we need to analyse this over several different time points to make sure that that is consistent over time. One of the areas that we're working on with colleagues in Glasgow is looking at the transmission models for this to try and use our data to better parameterise existing models, and essentially asking these three relatively simple questions, but using modelling to fill in some of the gaps which we can't collect from field data. We want to know where are the vectors and the hosts. We want to know how do they interact and how does this influence infection. And of course there's a temporal component in that, as I said before. So what we can do is use models to look at different environmental variables to see which are the most sensitive in predicting risk of infection, using our case control data. Because those cases and controls are selected to be matched environmentally, we can begin to look at that. And the idea here is on this plot, what you could do is look at these side on, and you would see as it becomes greener or more yellow, you would see, looking at it side on, you would see it rise up as it comes there. So they're laid on side by side there for information. And then we take those, and what this is saying, that spot there was the proportion of forest loss, and this one here is the proportion of core forest cover. So to me it's interesting that even from this kind of early predictive modelling, we're beginning to see the same patterns emerge as we saw from our broader scale village observations. That you've still got to have forest around, but the fact that some of it might have been recently cleared is both are important in that. And it's the spatial scales that seems to be important. And of course, you can, those of you au fait with spatial epidemiology will want to know, well, again, what about fragment sizes of forest? What type of forest are they? And we can begin to answer some of these questions. So then we move on to some of the simulations here, and this is some work that Paddy Brock's done. He's over there, and you can ask him the details. But effectively superimposing macaque movement patterns, informed, but not using our data, but informed from some of the data that we have on there, and then using some of the human movement data here. So we can see that actually it's heavily influenced by the type of movement. So humans is largely peri-domestic, except for those odd people that go off, whereas macaques is much more peripatetic and around where and they go, a lot more kind of variation. And so on those previous schema, we have our cases and controls in the red spots. We augment those by data from other houses who are not in that study, and we've just completed a large cross-sectional survey which will allow us to fill in those gaps. And so, by way of a climax, we'll show you how we think these all interact together, and this kind of old-school Space Invaders type graphic here, where we've got some mosquitoes in blue, some macaques in green, and some people in orange. And again, so these are parameterized by effects that we think are likely to happen. So this is mosquito emergence rates, all within credible levels of that we think are reasonable from our work. So you can begin to see infected mosquitoes arise. You'll soon see infected macaques, and then we will get some poor soul who gets infected with knowlesi towards the end. But the idea is to build this database increasingly so that these models become increasingly more accurate. It's going to be him there. Da-da. So that person has knowlesi. So, my summary. Integrated research and novel methodology are just applicable here. We've used knowlesi as a prominent health problem in this particular part of the world, but we've already been asked to look at, say, dengue in this same particular area because we have evidence. Well, dengue there is more rural and urban, so the suggestion of potentially some sylvatic transmission. But one can think of applying this to a number of different types of infection in areas where disease infection rates are low. And as you zoom down, and Dominic talked about foci this morning, where the disease gets rarer and rarer, you need more and more radical and detailed approaches to identify risk factors. The use of the new tools really is, it's not just fun, it is fun, but it allows us to really nail down that spatial interaction. And it is teaching us the best ways to look at that as well. And some of it will be overkill, we know that, but we have the ability to try and weed out what's giving us the best information. The drone in particular allowing us to do those frequent assessments of changing land use, changing vegetation. I've just been to Malaysia and it's experiencing the driest kind of period of time it has for the last 3 years, which I don't think is uncommon globally, but it is for them. And so we need to know how the environment is going to react to that. So we started some drone flights. And of course, it has regular effects, as I mentioned before, by the seasonality and fruiting, and then the land clearing, which is a dramatic, hopefully one-off effect. We, for our data, need to link this with infections in all the different components of the life cycle to really make it a plausible, dynamic model. And that work is ongoing now. So I am one of many, as all the speakers have been today, but I think the funders, as I say, have been really quite far-sighted in picking up this Living with Environmental Change initiative, and I'd like to thank everybody on that scheme. And thank you.
Questions, please?
So just sort of tying back into Dominic's talk earlier, where he talked about all the genetic data that's available and the ability to look at movement of parasites, essentially. Obviously you're pushing the boundaries already of the sort of technology and what you can infer. Is it completely out of the question that you might be able to attribute transmission from one group of macaques to one family and try and pin it down that way, exactly what's going on?
It's a very good question, and kind of obviously one that I anticipated and still don't have an answer for. It is something that we want to do. There are kind of practical and kind of ethical issues around that. We don't have the samples from the macaques that we want to have in terms of creating a kind of a background genetic kind of pool for knowlesi parasites. We do obviously have the parasites from the humans. Knowlesi is a very, very poor cousin to the other malarias, and that we've only got one reference genome at the moment. That will change. So at the moment we don't have too much information on likely variation of the parasite and where we would do all the approaches that David described this morning on those particular parasites. But it's certainly waiting in the wings to do that. And some people here Oxford are doing work on typing the macaques. And so there are preliminary evidence that certainly between species, the longtail and the pigtail, there are differences in infection, and that actually may be a physiological barrier to infection. So there will also be macaque level differences that we do need to figure in. And we have, if anybody wants them, a freezer full of feces of macaques, which we want to begin to type the macaques and look at familial sort of aggregation of infection within troops.
And you take part. Take it to the freezer.
It's the feces. It always gets them.
I'd just like to ask about GPS trackers.
Yeah.
And how easy was it to get people to use them? And where they have many instances of people just taking them off and leaving and forgetting about it?
The project has been very interesting in that respect, that human interaction. So most people have been really, really good about it. Most people are really interested because they... I don't know how many people have done social science research before, but you tend to have the first step, which is the focus group discussion, and you might get more voluble people in that group kind of taking over. And our experience, so we do this with something else called photo voice. So they have a GPS tracker, and actually sometimes the GPS is built into the camera, so when they take a picture we get a point of where they are. They like it because at least I can tell you what I'm doing now, so I really like it. You know, I can give you information about what I think diseases are, not all those and I talk to you one-on-one about what I think it is. So they quite like it as it's been coupled with that photo voice. We have had a few instances where, I haven't got it today, I gave it to my friend because they wanted to see what it was like to use it. So we've got several lines going off into the forest, and then one goes off to the market or the hairdressers or something like that. And then you think, hang on. So it's not 100% perfect, but it has been reasonably well. The uptake has been good and pretty solid. What I would say is very interesting about the project overall is that we have kind of suffered from our own, not popularity, but our own attempts to promote the project and to try and get community awareness to help us out. So now whenever we ask people a question about malaria, they always say monkey. That's the first thing they say is macaque, because we drive around with big cars with monkey bar on the side, and they just make this massive assumption. So in terms of trying to get to, on a social level, what do you think drive disease? They always say monkey. And all right, forget about the monkeys. What do you think? So it has been quite difficult we've contaminated essentially the project, but it's a kind of a difficult thing to have managed.
Just before we let you off, I've got one for you.
Okay.
What do you tell people locally about not getting knowlesi? What do you know about transmission to make you safer?
That is a very good question, because the clear, I'm a public health person. I want to, you want to make a difference here, and it's not just really nice epidemiology, which I think it is, but there has to be that vision at the end of it. I think it's really quite difficult. The message we have at the moment is all around self-protection and awareness. Yeah. Do you overnight there? You might have seen Kevin Baird's classic picture with that little, bloke climbing up a tree with a mosquito coil in a little kind of pan. So I mean, we those kind of things we're thinking about, because we do the vast majority of the risk is in people going into that forest. I think our concern is around those peridomestic cases, and that one we're a little bit kind of unsure about. Net use, the primary healthcare provision are pretty high in that particular area. So, I mean, all the other kind of basic building blocks of good malaria control are there. So knowlesi presents a really difficult challenge.